would like to start the scientific part of the program with our first speaker. Hopefully the slides will come up. Our first speaker is Dr. Enric Mateu. He's a professor of veterinary immunology and microbiology, I believe, animal health at the University Autónoma de Barcelona. He's been working with PERS pretty much his whole career. We won't say how long. So we're very honored to have him here to share with us a presentation on will genomics spell the end of vaccines. Enric?
Good morning. First of all, I have to admit that I was a little bit scared when Oliver called me and told me the title of the presentation, because indeed I know nothing about that. So it was pretty difficult for me to figure out what the title was meant to be, what this presentation meant to be. But after some talks and some conversation about that, I decided that, okay, maybe I can tell you something about this. And for those of you that don't want to listen to my talk, the short answer is no, probably not in the short term. But who knows in the long term if we can face something really, really new in the field. So, okay. So the final impact of PERS in a farm depends on a number of things. And obviously, the first thing is the likelihood of the virus arriving to the farm. Obviously, if the virus cannot reach your farm, you will not have the disease. But unfortunately, this is not the case for most of us. So the second thing is, okay, the virus is in the farm. Can this virus spread on the farm? Are we taking measures to avoid this dissemination within the farm? And that points, as we will see later, towards the internal biosecurity. And if we have the disease, obviously the final impact will be a sum of the individual outcomes of the infection in the animals that are infected. But not only this, also the disturbance of the flow of production. As Oliver said before, nowadays we are working in a system that needs to be very, very planned, very well programmed, with small variation, small diversity, because this permits this resilience thing. If we think that way, obviously, when we plan to control the infection in a farm, we normally talk about 4 pillars. Diagnosis and monitoring, mainly because that permit us 2 things. First, to have an early warning that our farm is being infected, and that's very important because that permits us to implement the measures as soon as possible. And second, because we can monitor the evolution of the infection in the farm. But we also need immunity, and the immunity normally in this field means the adaptation of sows, the acclimation of sows, recall vaccinations, and even piglet vaccination. We need biosecurity to reduce the likelihood of a new virus, a lateral introduction in the farm, but also to reduce the within-farm transmission. And obviously, we have to manage the herd. We have to control the flow of pigs in the farm to avoid the spread between different phases of the production. This is a very classical approach that we can summarize in this slide. By the biosecurity, we understand that we have an internal biosecurity, that is the mitigation of the spread of the disease within the farm. We have an external biosecurity, that is the number of measures that we can implement to avoid the entrance of the virus. We have management measures, we have the diagnosis, and the immunity. But we can look at this from a different point of view, from a complementary point of view. Indeed, the infection is the result of the interaction of the host, in this case with the virus, and this takes place within a given environment. And I will not talk about the environment. It's really important. You have other talks today about this. I will focus on the host and on the virus. What we can do with the host, obviously we can do two different things. They are not exactly the same. We can select for increased resilience, or we can create resistant peaks. And this is not exactly the same. There is a deep difference in the concept of the things that we are facing here. And both strategies are now in the focus of many companies because both things are promising in the sense of controlling PERS. So again, definition. Today is the definition day, okay? Resilience, the capacity to recover quickly from difficulties, toughness. And I think that this is very, it's very plastic, this definition of the toughness. You can survive well to the problems, okay? And this is I dare to say that this is now a classical experiment to show how we can improve resilience. In this work, the authors had a very large group of animals, and there were 3 groups, about 565 pigs that were infected with PERS2, and they were followed up during the infection. This is the graph showing the evolution of the viremia. As you can see here, most of the pigs had a 21 or 28 was the most common viremia. And then afterwards, you can see some rebounds of the viremia that probably were not related to the genetic background of the animals because no statistical correlation with that could be found. The interesting thing here is that you can see that in spite that most of the animals end the viremia by day 28, not all animals had exactly the same behavior. In other words, the viral load that they were carrying for the long time of infection was not exactly the same. And the case is that when you look at the body weight of those animals, the situation is similar. This is the body weight of the animals, and obviously some low weight at the start of the infection were low weight, but then you have all other situations in the middle. And obviously the question was pretty clear at this point. Is there any correlation between this viremia, this body weight, and can this be correlated to the genetic background of the animal? And we will see here. Investigating regions of the genome that could be associated with better or worse weights, with better or worse viral loads, measured as the area under the curve for the whole period of infection. They found 2 regions they found one chromosome 4, here and here, associated with the viral load and the body weight gain. And these markers in the chromosome 4 explain 15% of the variance for the viral load and 11% of the variance on the body weight. So that was promising. In chromosome 4, there is something that can be related to this resilience. And that's proof. When they look at the genotype of those animals, this is the viral load. To the right, more virus. This is the body weight. This sends more body weight, okay? So these animals here that were AA for one of these markers had lots of virus during the infection and low body weight. These animals here that were BB, less virus, more body weight. And in the middle, AB, not good as this one, not bad as this one. So at this point, it was pretty clear that you can increase the resilience of your animals just by doing a classical selection based on some SNPs. In this other work, the approach was similar, but in this case using vaccination. The intention of the work was to see what happened with regards to the genotype of the animals when you vaccinate the animals and then do a challenge of those animals for PERS, but also for PCV2. And I would like to show you what happened regarding the average daily gain. These are the animals. In this section, you have PERS. This is the co-infection period. And you can see again we have AA and AB. AA and AB. No difference in this moment. But if we look at what happens for PERS or for PCV2 separately, you can see here some differences between the non-vaccinated, some difference between the vaccinated with regards to the viral load. And in the case of PCV2, this was pretty clear. Both genotypes, AA versus AB, were not having the same behavior. So that means that also this answer related to the vaccination and to the development of the infection after the vaccination was not exactly the same. In other words, we can improve the response to vaccination by means of the genetic selection. Similarly, with other markers, and you said before that for many years had been said that pure breeds are performing worse, okay? And I think that's the case. And if you look here, for example, when they look at the Duroc versus large white versus a crossbred large white, you can see again AA in this case versus AG. There were differences between breeds, but also differences for the genotype. So that means that taking a very classical approach of genetic selection, you can improve the response of your animals against infection. But resilience can be also improved by other means, not only by genetic selection. And I would like to show you a couple of papers, the results of a couple of papers dealing with adjuvants. And why I'm putting these adjuvants here is because adjuvants are not genomics, of course. Just because I think that this is a field that has to be explored. For many years, we have been said that adjuvants had no effect on the immunity against PERS. This has been published some years ago, but I think this is not the case. I think that the use of adjuvants can improve the response. In this paper, they use carbopol as adjuvant to improve the response of the modified live vaccine against PERS. Very classical approach, one control with the adjuvant, the vaccine alone, the vaccine plus the adjuvant. And just to show you one result, 14 days after vaccination, the group receiving carbopol, half of the animals had an important response for the production of interferon gamma as measured in the interferon gamma ELISpot. And what I would like to bring to your attention is this number here, 14 days. Because one very important thing for us in the farms is that in spite that we can perform vaccination, we can improve biosecurity, the virus is in there. And the higher the infectious pressure in the farm, the lower the window that we have to protect the animals. So to have a very early response, it's pretty important to narrow this window of infection within the farm. The second one is a completely different thing, but again, using adjuvants. And in this paper, they use an experimental inactivated vaccine. And I would like to bring to your attention here, these were inactivated. Because for many years, we have been saying the inactivated vaccines are not efficient for this. And the case is that probably they are not efficient for the primal vaccination of the animals, but we can also improve the performance of these inactivated vaccines. In this work, these people use two control groups, one adjuvant, no PERS, so this is a mock vaccinated group. Then they had two groups in which the inactivated vaccine was used with one of the adjuvants. It's not important at this point, the type of adjuvant here. And two other groups with a different adjuvant, a classical oil adjuvant, okay? They were subjected to a double vaccination, and they were challenged with either type 1 or type 2 PERS. And the results here show that blue and red, these are the mock, develop a nice viremia, classical viremia. These two groups here, orange and black or gray, here were the oil adjuvant, with the vaccine. They had some level of protection, but very low. And these groups were, green and violet, were the groups vaccinated with the other adjuvant, with the Montanide adjuvant. So they were partially protected, but much better than these groups here, okay? And this is the same. This is by PCR. This is by isolation of the virus. Pretty much the same. Violet and green, orange and gray, blue and red that are the controls. So just by using different adjuvants, you can increase the immunogenicity, the performance of your vaccinated animals. And this is a word that probably should be explored in the next years. Okay, but we have a different side of this tale. We have the resistance. What is resistance? Resistance is a force that acts to stop the progress of something or to make it slower. This is the definition of the dictionary. And if we talk about resistance against PRRS, we are talking about resistant peaks. And to understand how these resistant peaks are made, we have to look at the pathways of entry of the virus. This is taken from this paper and tries to show the way in which the virus enters in the cell. This is the virus, this is the macrophage, and we have first a number of receptors in the cell surface that interact with the outer proteins of the virus. Normally we talk about heparan sulfate as the unspecific receptor. For type 1 sialotezenes have been shown to be some of the potential receptors, but in the end, we need the contribution of this green molecule here that is CD163. Without the intervention of CD163, there is no decapsidation, there is no start of the replication, so CD163 is thought to be the essential receptor. So the idea is, as a concept, is quite simple. If we have peaks with no CD163 expression, knockout peaks, obviously there is no ground for the infection. And in a brilliant piece of work, they demonstrated, Kristen Whitworth and co-workers demonstrated that this is possible. They edited peaks in the CD163 just to make knockout peaks and see. CD163 positive animals infected, a nice viremia, a nice development of antibodies, CD163 knockout peaks, no viremia, no antibodies, no infection. So in other words, with these peaks that have been manipulated to be CD163 negative, the infection did not take place. But when we are modifying something, when we are modifying the expression of a molecule such as CD163, that is an haptoglobin receptor, is involved in the iron physiology in the organism, maybe we are doing some other things. And here there are this, I recommend you to read this paper about this thing. It's pretty simple, but very well written. And there are a number of concerns about this. The first concern is about the legal and regulatory aspects of this. Indeed, we are manipulating an animal, and obviously we want to have, the people that are doing that want to have a patent for that, okay? And this is maybe in the United States is more clear, in Europe is not that clear, this thing. The second, what is the efficiency of these pigs when they are put in the farm? Obviously, let's imagine that there is something that only will affect 100% of the edited pigs. You will notice that after putting hundreds of thousands of pigs in the field. So there is a ground for improving here, but we need still lots of information. What are the side effects of knocking out CD163? If the pig is challenged by other pathogens, what will happen? Who knows? This has to be tested. And obviously, there is another point about the stability of CD163 deletion that is the most easy to answer because probably these animals will be stable. Okay, but we can look at this improvement from the other side. The host and the virus. And if we look at the virus, this is a representation of pairs, just pretty simple representation. But it wants to show you that this virus is composed by the number of proteins that are in the surface that form part of the virion, that are called the structural proteins, the proteins that you can find in the viral particle. And then you have some proteins that are produced only during the replication cycle, that is what we call the non-structural proteins, okay? This virus replicates because it encodes for an RNA polymerase, and the interesting thing here is that this polymerase is not very, precise. It commits lots of errors. And this makes that every time that the viral genome is replicated, some mutations are introduced. Obviously, not all mutations will be fixed in the genome. Some of the mutations will be little, will produce that the viral particle cannot infect the cells or cannot replicate, so these little mutations will disappear from the population. Some other mutations are neutral. You know that the genetic code is degenerate. That means that different codons can code for the same amino acid. Or some of the mutations may be fixed because they provide some gain to the fitness of the virus. As a result of this, the virus is present as a viral quasi-species. That means that indeed in the animal, you have lots and lots and lots of variants of the same virus. Each viral particle is harboring some mutation, some change that can be important or not for the evolution of the virus. So in other words, in the animal, the virus has diversity. Not all viral particles are exactly the same. And this is important. It's like, I always put the same example. You can think of a hummingbird. Do you realize how a hummingbird lives? How is a hummingbird life? Hummingbird only, you know what is a hummingbird? Colibri in Spanish. I don't know any other word to say. It's a small bird that can only live by eating on a given orchid. Normally it's an orchid, taking the pollen of an orchid. And the level of, of evolution and specialization is that big that most of the hummingbird species can only feed on one species of flower, okay? So that means that this miracle of the nature will disappear just by increasing the temperature 1 or 2 degrees because this orchid will disappear. And then we have human beings. We can live everywhere, from the pole to the desert. We are able to adapt because we can be very resilient. The hummingbird is not. That's the idea. So with the virus, it happens the same. When you have diversity, the virus can adapt to the different situations. The more the pressure you put there, the diversity decreases. And if the diversity decreases enough, the virus collapses. The population of the virus collapses and disappears. And this viral evolution, this diversity, obviously, is not infinite. This diversity has limits. Not everything is possible because you need a virus that can replicate, can be transmitted, and should be functional. And this is what I want to show you. For HIV, it's known in the last 4 or 5 years that there is what is called a transmission bottleneck. What is a transmission bottleneck? When the scientists look at the virus that is in the people that are infected, in comparison with the virus that reach a new infection, and this has been studied in partners, in sexual partners, they realize one thing. The donor person, the person that is infected and is transmitting the infection, has a huge variability of virus in the body. But the one that is infected, the recipient of this infection, only is infected by what is called a founder variant. Only very few of the variants present in the donor are able to cross this barrier, that means the sexual mucosa in this case, to establish infection. And this is a concept that is somewhat revolutionary. Because if you think of what is the minimal infectious dose, we say, for getting the infection in an individual, we have to permit a minimal number of particles reaching that mucosal surface. This phenomenon points to a slightly different thing. It's not a question of how many particles. It's a question of how many particles can pass through this bottleneck. And this is not exactly the same, because if you think a little bit about that, that means that the mucosal barrier only has to have immunity, I mean, a specific immunity, against those variants that can cross the barrier. All the other variants will be kept in the barrier with or without a specific immunity. You get the idea, more or less? So, since we have free time to do these things, we decided to examine this bottleneck in pairs. And the experiment was pretty simple. We took some naive pigs, 9 pigs in this case, and we inoculated these pigs with a PRRS1 isolate, but the intranasal route that is more or less a common route of infection in the experimental model. In the second experiment, what we did was we took animals that were infected, and then we put in contact these animals with other naive pigs, just to see what happened in the experimental transmission versus the natural transmission. Okay, in this case, we had 5 events. So we took the pig sera every 2 days, just to be sure of the period of transmission, and we examined the diversity in the viral particles by means of a process that is called the next generation sequencing. In other words, we took the virus, we replicated just one time in macrophages to have enough virus to be sequenced, and we examined 1,000 reads for each position. Okay, so we had, for a number of viral particles, 1,000, what is the nucleotide that we have in this position all along the genome, okay? And that's the first result. This is the diversity in our inoculum. This 0.013 means that 1.3% of the positions in the genome of the virus has 15,000 nucleotides, okay? So in 1.3% of these positions, we can find viruses harboring different nucleotides, okay? This is the diversity that we have here. When we infected the pigs with this inoculum, look what happened. All but one, at day 1 of viremia, had less diverse virus than the diversity that we have in the inoculum. So that means that the number of changes that we see in the genome of the virus decrease after the inoculation, right straight after the inoculation. And that was promising, okay? But maybe we are putting the virus by a way that is, creating some artifact, because this is an experimental inoculation. So the next thing was to assess what happened with the natural transmission. And look, again, except in one case, the diversity decreased, and decreased very seriously. This virus here, for example, or here, were almost with no diversity. So that means that apparently a transmission bottleneck occurred. We are working to define exactly what positions are the most relevant here, and this is a serious bioinformatic work, so I don't have the results yet. And the next question was, there is a bottleneck caused by immunity, because this can give us clues about how to improve vaccines. So we took animals, again, that had partial immunity because they had been immunized before, and we studied 11 transmission events under natural conditions. One infected animal is penned together with one of these animals that had been vaccinated some weeks before. What we saw is that of these 11 pigs, 7 pigs developed a viremia of about 1 week, between 7 and 14 days, more than 1 week. And 4 pigs developed a viremia for less than 5 days. That means 1, 3, or 5 days, okay? And the interesting thing here, going back to the neutralizing antibodies, is that the longer viremias correlated with the animals with titers below 1 to 8, and the shorter viremia is correlated with the titers 1 to 16. So more or less, we are on the line of what we were told yesterday. The interesting thing, when we look at the virus in the animals that were infected, in these 11 animals that were infected, it was pretty clear that the animals with the short viremias were not equal to the animals with the longer viremias. Do you remember the last slide about the diversity? Yes? What we had is that for all animals, I mean, when you compare 7 versus 5, okay, versus 4, sorry, for all animals, there were differences related to the group. And these differences occur only in 55 positions of the genome, of which 37 were here and 9 were here. And this is not at random. Obviously, if you have 15 kilobases, it's impossible just to have 55 changes concentrated in 2 proteins of the virus. And that means that most of the changes were concentrated here. This is the viral polymerase. And the other number of changes were concentrated here in the gene encoding for the GP2, that is one of the proteins related to the entrance of the virus in the cell. So we still lack the precise significance of these results, but I can advance from yesterday night that, at least from the prediction models, these changes here in the replicase imply a change not only in the sequence of the replicase, but also in the 3D conformational structure of the replicase, in the point where the RNA enters the replicase to be replicated. So that means that there is something important here. So take-home messages. One, about the resilience. We can improve the resilience by classical ways, and we know how to do that. So use it. Second, adjuvants are useful. This is not genomics, but I want to emphasize that. Adjuvants are important. Maybe you can take a look deeper on that. About the resistant peaks, this is the future probably. But we don't know yet the performance of these peaks in the field. And this is really important because this business is about the performance of the animals in the field. 4, the bottlenecks that probably we're seeing suggest that there are some points in the genome, some points in the virus that are critical for allowing this crossing the mucosal barrier. So this is worth to be studied more. And from the second study that we did, the clearing of the viremia was the classical example that is put as a catastrophic selection bottleneck. You are selecting for variants that are able to live in that aggressive environment, but by doing that, you decrease diversity, and by doing that, you produce a catastrophe of the population because you are decreasing the fitness, indeed, of this population. And I think that's all. Thank you very much.